Welcome to our exploration of one of nature's most remarkable processes, photosynthesis. This fundamental biological process sustains nearly all life on our planet. Photosynthesis is the process by which plants, algae, and some bacteria convert light energy from the sun into chemical energy. In this process, sunlight is captured and used to transform carbon dioxide and water into glucose and oxygen. Photosynthesis forms the foundation for nearly all life on Earth. It produces the oxygen that most organisms need to breathe and creates the energy-rich compounds that sustain the food chain. In ecosystems, photosynthesis makes plants the producers in the food chain. Plants create their own food, while animals are consumers that depend on plants either directly or indirectly for energy. To summarize what we've learned about photosynthesis, photosynthesis converts light energy to chemical energy, produces oxygen and glucose, forms the foundation for nearly all life on Earth, and makes plants the producers in food chains. At the heart of photosynthesis is a fundamental chemical reaction. The simplified chemical equation is 6 carbon dioxide plus 6 water molecules plus light energy yields 1 glucose molecule and 6 oxygen molecules. The reactants include carbon dioxide from the atmosphere, water absorbed through plant roots, and light energy captured by chlorophyll. The products are glucose, an energy-rich sugar molecule, and oxygen, which is released as a byproduct. This seemingly simple equation represents a complex series of chemical reactions that transform inorganic compounds into organic compounds. The energy from sunlight is captured and stored in the chemical bonds of glucose molecules. Plants can later break these bonds to release energy for various cellular processes. This chemical reaction is the foundation of nearly all life on Earth, converting inorganic carbon into organic molecules that support the entire food web. As we delve deeper into photosynthesis, we'll explore the plant structures that make this remarkable process possible. Plants have evolved specialized structures for photosynthesis. These include leaves that capture sunlight, chloroplasts where the chemical reactions occur, stomata for gas exchange, and roots that absorb water and minerals. Chloroplasts are specialized organelles that serve as the primary site of photosynthesis. Inside the chloroplast is a fluid-filled region called the stroma, where the light-independent reactions occur. Embedded within the stroma are flattened membrane sacs called thylakoids, which contain chlorophyll and other pigments that capture light energy. Thylakoids are often arranged in stacks called grana. These structures increase the surface area for light absorption and are where the light-dependent reactions take place. Leaves are the primary photosynthetic organs in plants. Their structure is highly specialized to maximize photosynthetic efficiency. The upper epidermis is transparent to allow light to enter. Below it is the palisade mesophyll, which contains cells densely packed with chloroplasts. The spongy mesophyll layer has loosely arranged cells with air spaces that facilitate gas exchange. The lower epidermis contains specialized openings called stomata. Stomata allow carbon dioxide to enter the leaf for photosynthesis and oxygen to exit as a byproduct. The leaf's overall structure is designed to maximize light capture with its broad, flat shape, while facilitating gas exchange and housing the chloroplasts where photosynthesis occurs. While photosynthesis occurs primarily in the leaves, roots play a crucial role by supplying the necessary water and minerals. The root system consists of a main taproot with lateral roots branching outward, maximizing the area for absorption. Tiny root hairs vastly increase the surface area for water and mineral absorption from the soil. The water absorbed by roots is essential for the light-dependent reactions of photosynthesis, where it is split to provide electrons and hydrogen ions. Minerals like magnesium are used to build chlorophyll molecules, while nitrogen is needed for the enzymes that catalyze photosynthetic reactions. Chlorophyll is the primary pigment responsible for giving plants their characteristic green color. Chlorophyll is a complex molecule with a ring structure containing a magnesium atom at its center. This structure is crucial for capturing light energy during photosynthesis. Chlorophyll primarily absorbs blue and red wavelengths from the visible light spectrum. It reflects green light, which is why plants appear green to our eyes. The absorption spectrum of chlorophyll shows strong peaks in the blue region around 430 nanometers and in the red region around 662 nanometers. While chlorophyll is the primary pigment, plants also contain various accessory pigments that capture light energy at wavelengths where chlorophyll is less efficient. Accessory pigments like carotenoids, xanthophylls, and phycocyanins absorb light in the green, blue-green, and other regions of the spectrum that chlorophyll cannot effectively capture. Together, these pigments ensure that plants can utilize a broader range of the light spectrum for photosynthesis, maximizing their energy capture efficiency. The light-dependent reactions form the first major stage of photosynthesis. These reactions occur specifically in the thylakoid membrane of chloroplasts. The process begins with two specialized protein complexes called photosystems. When light strikes the chlorophyll molecules in these photosystems, it excites electrons to a higher energy level. These excited electrons are channeled through an electron transport chain, moving from photosystem 2 through a cytochrome complex to photosystem 1. As electrons flow through the transport chain, their energy is used to pump hydrogen ions into the thylakoid lumen. These ions then flow back through ATP synthase, generating ATP. At the end of the electron transport chain in photosystem 1, the electrons are used to reduce NADP+ to NADPH, another energy carrier. To replace the electrons that have been transferred from photosystem 2, water molecules are split in a process called photolysis. When water is split, oxygen is released as a byproduct. This is the source of nearly all the oxygen in Earth's atmosphere, making photosynthesis essential for aerobic life forms, including humans. To summarize, the light-dependent reactions convert light energy into chemical energy in the form of ATP and NADPH, while releasing oxygen as a byproduct. These energy-rich molecules, ATP and NADPH, will power the next stage of photosynthesis, the Calvin cycle. The Calvin cycle, also known as the light-independent reactions, is the second major stage of photosynthesis. It takes place in the stroma of chloroplasts and uses the energy carriers from the light-dependent reactions to convert carbon dioxide into glucose. Unlike the light-dependent reactions which occur in the thylakoid membranes, the Calvin cycle occurs in the stroma, the fluid-filled space inside the chloroplast. The Calvin cycle requires several key components: carbon dioxide from the atmosphere, ATP and NADPH from the light-dependent reactions, and the enzyme rubisco, which catalyzes carbon fixation. The Calvin cycle is a circular process consisting of three main steps: carbon fixation, reduction, and regeneration. In step 1, carbon fixation, the enzyme rubisco captures carbon dioxide and attaches it to a 5-carbon compound called RuBP, forming an unstable 6-carbon compound that splits into two 3-carbon molecules. In step 2, reduction, ATP provides energy and NADPH provides electrons to convert the 3-carbon molecules into a compound called G3P, or glyceraldehyde 3-phosphate. In step 3, regeneration, some G3P molecules are used to regenerate RuBP, the carbon dioxide acceptor, so the cycle can continue. This requires additional energy from ATP. For every 6 carbon dioxide molecules that enter the cycle, 1 glucose molecule is produced, while consuming 18 ATP and 12 NADPH molecules from the light-dependent reactions. The Calvin cycle is named after American biochemist Melvin Calvin, who discovered the pathway in the 1950s. Calvin and his colleagues used radioactive carbon-14 to trace carbon's path through photosynthesis. The Calvin cycle plays a crucial ecological role. It removes carbon dioxide from the atmosphere and incorporates it into organic molecules, a process known as carbon fixation. This process is the foundation of nearly all food chains and provides oxygen as a byproduct of the overall photosynthetic process. Understanding the Calvin cycle helps us appreciate how plants convert carbon dioxide into the sugars that sustain life on Earth. Photosynthesis is affected by several environmental factors that can enhance or limit its efficiency. These key environmental factors include light intensity, carbon dioxide concentration, temperature, and water availability. Light intensity directly affects the rate of photosynthesis. At low light levels, photosynthesis increases proportionally with intensity. In the light-limited region, providing more light increases photosynthetic rate. However, at higher intensities, the process becomes light-saturated, where other factors such as carbon dioxide or enzyme availability become limiting. Carbon dioxide concentration is critical for photosynthesis as CO2 is a key substrate for the Calvin cycle. Increasing CO2 concentration typically enhances photosynthetic rate until other factors become limiting. Current atmospheric CO2 levels are often suboptimal for maximum plant growth. Temperature affects photosynthesis because it influences enzyme activity. Each plant species has an optimal temperature range. At low temperatures, reaction rates slow down. As temperature increases, photosynthesis increases until it reaches an optimal point. Above the optimal temperature, enzyme denaturation begins to occur, reducing photosynthetic efficiency. Water availability significantly impacts photosynthesis through several mechanisms. When water is abundant, stomata open, allowing carbon dioxide to enter the leaf while oxygen exits. This facilitates efficient photosynthesis. During water stress, stomata close to conserve water, but this also restricts carbon dioxide intake, limiting photosynthetic rate. The concept of limiting factors is crucial for understanding photosynthetic efficiency. According to Liebig's law of the minimum, the scarcest resource determines the overall rate of photosynthesis. Like water in a barrel with staves of different heights, photosynthesis can only proceed at the rate allowed by the most limiting factor. In this example, water availability is the limiting factor. Plants have evolved diverse adaptations to optimize photosynthesis in different environmental conditions. There are three main photosynthetic pathways, C3, C4, and CAM. Each offers advantages in specific environments. C3 photosynthesis is the most common pathway, found in plants like rice, wheat, and most trees. It's efficient in moderate climates, but struggles in hot, dry conditions. C4 plants like corn and sugarcane have evolved a specialized anatomy that concentrates carbon dioxide around rubisco, reducing photorespiration and improving efficiency in hot environments. CAM plants such as cacti and pineapples open their stomata at night to collect carbon dioxide, storing it for use during daylight. This dramatically reduces water loss in arid environments. These diverse adaptations demonstrate how plants optimize photosynthesis in response to environmental challenges, ultimately ensuring their survival and success across different habitats. Photosynthesis plays a crucial role in the global carbon cycle. The carbon cycle describes the movement of carbon between the atmosphere, oceans, soil, and living organisms. Plants are essential components of this cycle, acting as carbon sinks through photosynthesis. Plants act as carbon sinks by absorbing carbon dioxide from the atmosphere during photosynthesis. This carbon is then converted into glucose and stored in plant tissues, effectively removing it from the atmosphere. Photosynthesis and cellular respiration work in a delicate balance to maintain atmospheric carbon dioxide levels. Plants absorb carbon dioxide during photosynthesis and release it during respiration, creating a natural equilibrium. Human activities are disrupting this natural balance in several ways. Deforestation reduces the Earth's capacity to absorb carbon dioxide by removing trees that act as carbon sinks. Meanwhile, burning fossil fuels releases carbon dioxide that has been stored underground for millions of years. These activities combined are increasing atmospheric carbon dioxide levels at an unprecedented rate. The resulting carbon imbalance is leading to increased levels of carbon dioxide in the atmosphere. Atmospheric carbon dioxide has risen from about 315 parts per million in 1960 to over 415 parts per million today. This increase is driving climate change, resulting in rising temperatures, sea level rise, extreme weather events, and ecosystem disruption. Understanding photosynthesis and the carbon cycle is crucial for addressing climate change and developing effective carbon management strategies. Photosynthesis first evolved approximately 3 billion years ago in ancient cyanobacteria. To understand its significance, let's look at a timeline of Earth's history. Around 3 billion years ago, cyanobacteria developed the ability to perform oxygenic photosynthesis, using water as an electron donor and releasing oxygen as a byproduct. This innovation was one of the most transformative events in Earth's history, dramatically changing our planet's atmosphere and enabling complex life to evolve. The evolutionary innovation of oxygenic photosynthesis led to what scientists call the Great Oxygenation Event. Before photosynthesis evolved, Earth's atmosphere was dominated by reducing gases like carbon dioxide, methane, ammonia, and hydrogen sulfide. As cyanobacteria produced oxygen through photosynthesis, it initially reacted with dissolved iron in the oceans, forming iron oxide deposits. Eventually, oxygen began to accumulate in the atmosphere. Between 2.4 and 2.0 billion years ago, atmospheric oxygen levels rose dramatically, transforming Earth's reducing atmosphere to an oxidizing one. This oxygen-rich atmosphere had profound effects on life's evolution. It enabled aerobic respiration, which produces more energy than anaerobic processes. This facilitated the evolution of complex multicellular organisms. Additionally, oxygen in the upper atmosphere formed the ozone layer, which blocks harmful ultraviolet radiation, allowing life to colonize land surfaces. Without photosynthesis and the resulting oxygen, complex life as we know it today would not have been possible. Another major evolutionary milestone was the origin of chloroplasts through endosymbiosis. According to the endosymbiotic theory, chloroplasts originated from free-living photosynthetic bacteria that were engulfed by early eukaryotic cells. Instead of being digested, these bacteria formed a mutually beneficial relationship with the host cell. Over time, the engulfed bacteria lost their independence and became organelles, what we now know as chloroplasts. This symbiotic relationship proved highly beneficial. The host cell gained the ability to perform photosynthesis, while the engulfed bacterium gained protection. Evidence supporting this theory includes the fact that chloroplasts contain their own DNA and replicate independently within the cell, similar to bacteria. A similar endosymbiotic event is believed to have given rise to mitochondria, the organelles responsible for cellular respiration. The endosymbiotic origin of chloroplasts represents one of the most important evolutionary innovations in the history of life on Earth, setting the stage for the evolution of plants and all organisms that depend on them. Photosynthesis research provides remarkable applications in agriculture, renewable energy, and environmental conservation. In agriculture, scientists are engineering crops with enhanced photosynthetic efficiency. The C4 Rice project aims to introduce more efficient carbon fixation mechanisms into rice, potentially increasing yields by up to 50%. The RIPE project is engineering plants to recover faster from photoprotection, helping to increase global food security for our growing population. In renewable energy, researchers are developing artificial photosynthesis systems that mimic plants' ability to convert sunlight into chemical energy. These technologies use specially designed catalysts to split water into hydrogen and oxygen, creating clean fuels. These systems could potentially produce zero-emission hydrogen fuel, offering a sustainable alternative to fossil fuels. For environmental conservation, researchers are developing enhanced carbon capture systems based on photosynthesis principles. These include optimized forests, algae-based systems, and biomimetic artificial leaves designed specifically for CO2 sequestration. Interestingly, algae can be up to 10 times more efficient at capturing carbon dioxide than trees, making them particularly promising for climate change mitigation. Looking to the future, photosynthesis research faces important challenges, including scaling technologies from laboratory to industrial levels, improving efficiency, and addressing the costs of implementation. However, promising opportunities exist through interdisciplinary research, advances in synthetic biology, development of new catalyst materials, and artificial intelligence-driven discoveries. Photosynthesis remains one of nature's most remarkable processes, inspiring scientific innovation and offering potential solutions to some of our most pressing global challenges.